We have the pleasure today of having a second awardee presentation. So the ASLO presents the John H. Martin Award to one paper each year that has led to fundamental shifts in research focus and interpretation of a large body of previous observations. The 2021 John H. Martin Award is for Viruses and Nutrient Cycles in the Sea by Dr. Stephen Wilhelm from the University of Tennessee in the United States and Dr. Curtis Suttle, my apologies, from the University of British Columbia in Canada. The viral shunt is now a fundamental concept in aquatic sciences. The idea originated in this important paper. In short, the viral shunt is a term used to describe the phenomena in which viral infection of marine microbes redirects organic carbon flow away from higher trophic levels and through the microbial loop. This paper brought about an important conceptual shift. At a time when much of the research was focused on describing viral diversity and distribution, this article pointed to the now well understood biogeochemical importance of viruses. The calculations presented in this paper showed that a staggering 25% of all photosynthetically fixed carbon and associated nutrients in the ocean may be recycled through the viral shunt. Additionally, Wilhelm and Suttle made some of the first estimates of the total carbon stored in the viral pool and made eye-opening comparisons to the carbon stored in other types of marine organisms. The foundational research communicated in this article has since sparked empiricists and modelers to include viruses in efforts to understand nutrient and carbon flow through marine and freshwater systems. Today, the concept of the viral shunt continues to influence our understanding of ocean ecosystem functioning and nutrient cycles. With more than 1,000 citations, where about a third of these were within the last 5 years, the legacy of viruses and nutrient cycles in the sea and the viral shunt concept is still strongly felt in the field today. As the nominator notes, it is rare that an entire level of biological organization emerge from the shadows to become key components of science. But this seminal article did just that for viruses, bringing the idea of the viral shunt and the implicit role of viruses in marine carbon cycles to the aquatic science community. Please join me in congratulating and recognizing our 2021 John H. Martin awardees, Drs. Wilhelm and Suttle.
Thank you very much. We're on here. I will go ahead and share screen. Okay, so thank you, Amina. Thank you to the committee. Thank you to ASLO. ASLO's been in my home in science for 30 years, so this is pretty fun. But we're going to take a walk down memory lane here, and I want to tell you a little bit about what we were thinking when we wrote this paper. I'll talk to you a little bit about what's happened with viruses in the last 20 years, and then maybe give you some insight on where we're going. So what you're looking at here, of course, is some pictures from the Wayback Machine of what the group looked like when I was a postdoc with Curtis at the University of Texas and at the University of British Columbia. And it wouldn't be a talk with Curtis Suttle as a co-author unless I started with one simple reminder. And that is, if I can get my slides to go forward, that the ocean is full of viruses. There are literally hundreds of thousands of viruses in every drop of seawater. And I changed one of Curtis's classic slides to add in lake water too, because many people in my research group study lakes now. And this was sort of the foundation of what we were thinking about in the 1990s. We also knew that viruses were very fragile. And if there's a large number of viruses and they turn over very quickly or they're destroyed very quickly, they have to be replaced fairly quickly. And our thinking is that this replacement would lead to cell lysis and cell death. So we wrote the paper between 1995, when I started as a postdoc with Curtis, and 1999, when I took my position here at the University of Tennessee. We were asking very basic questions at the time. As you can see, we were looking for all sorts of things, but we just really wanted to know how abundant viruses were and if they're everywhere. Who do viruses interact with in nature? And how do viruses influence productivity? How do they influence carbon flow? The sort of central part of the viral shunt is this diagram here. And this diagram was an assignment that I more or less received from Curtis to try to figure out what was constraining our experiments. What were the bookends on measurements we might figure out? And we estimated that between 6 and 26% of photosynthate coming into the system may be diverted away from the grazing food web back into this viral shunt. I want to point out at this point that we're not the only ones who are thinking along these lines. There were a large number of researchers back in that time that we interacted with closely. We interacted with people like Jed Furman. I wanted to put one of Jed's models up here because he was really thinking forward there too. His student, Rachel Noble. But then also Karina Broussard, Greg Stewart was a student with Farouk Azam. Our friends in Norway, Gunnar Brackback, Mikkel Heldal, even the current ASLO president was working on viruses in that period of time. So the community was really thinking about and interacting very positively on what viruses may be doing in natural systems. And I think that this positive interaction has maybe been some of the best parts of having done this work, our ability as a group to come together and to talk. So what have we learned really since we wrote this paper and talked about carbon flow? Well, one thing we learned is that viral activity isn't just about carbon. Viruses drive nitrogen cycles, viruses drive phosphorus cycles. When I set up my lab at the University of Tennessee, because I had done my PhD with Charlie Trick, looking at iron availability in cyanobacteria, I was very interested in looking at the role of viruses in recycling iron. We did a number of studies, one highlighted here by PhD student Leo Corvin, a collaboration with Dave Hutchins, who was at the University of Delaware at the time, where we were able to show that viruses could be driving significant iron recycling to supply all the eukaryotic iron demands. This wasn't just a model. Leo actually went through and made all the empirical measurements along the way to back this up. So we really were able to show in the last 20 years that viruses are important. They are helping with these systems. One other thing we learned is that lysates are not always dissolved. Lysates are a continuum of dissolved to particulate organic matter, and that our thinking prior to this paper might not have been as accurate as it could have been. I was thinking at least that everything went into the dissolved size class. One other thing we've learned along the way is that there's a lot of spatial variability in virus activity, and the factors that constrain this spatial variability are different in each unique location in the ocean. The example I give here is some work by Janet Rowe, who was a PhD student who went to multiple oceanographic regions using the same tools, the same techniques, and the same pair of hands to measure virus production. And when she was able to do this, she was able to see that in different parts of the ocean, different factors influence virus activity. So what's new and maybe surprising about viruses? I think the most surprising thing for me, I think Curtis feels this way too, is the growth in the community. I was lucky enough to attend the Ocean Sciences meeting in 1996. There were 12 people in the room for the virus session. It's a very small, tight-knit group. At this ASLO meeting, 2021, there have been 5 sessions on aquatic viral ecology. Thank you, Karina, and thank you, Dolores, for putting these together. If you search the abstract books, you find 15 other sessions where viruses are at least mentioned. We even have our own meetings now. Next week, we'll be gathering virtually again for the Aquatic Virus Workshop, which has 190 registered participants who are interested in how viruses influence life in freshwater and marine systems. We've learned a few other things, though. In the late 1990s, when we wrote this paper, at least I was of the opinion, as were others, that most of the viruses in aquatic systems were phage. But we now know that large double-stranded DNA viruses, the giant viruses, as well as small RNA viruses, are really abundant. In fact, from some work we've been doing, transcripts suggest that viruses infecting eukaryotes may be more active than viruses infecting phage. We know that viruses just aren't little simple structural material blueprints. Bacteriophage, and especially the giant viruses, carry genes that are historically thought of to be cellular. We call them auxiliary metabolic genes. The great example is genes for photosynthesis in cyanophage. And our newest argument is that viruses may have metabolism. In looking at the proteins that are packaged inside the virus that's spinning around here on the left, the Oriococcus anophageferens virus, we find carbohydrate lyases, proteins that when released can work to break down carbohydrates. This means that when we're looking at dissolved enzymes, we may in some cases simply be looking at enzymes that are packaged inside virus particles. I think there's other new things on the horizon. Amina, when you called him Professor Suttle, I think that was a Freudian slip. Maybe you'd seen this. In 2017, I was lucky enough to write a paper with Matt Sullivan and Joshua Weitz where we talked about the viral shunt, or what we called the viral shuttle. And we talked about how virus activity may keep nutrients in the water column, or it may help export things. A lot of that depends on where lysis, or where infection happens in the water column. It also depends potentially on who's infected, as we saw by a recent paper and summary that was in Nature Geosciences. I think then moving forward, what's on the horizon is really going to be a better understanding of how viruses fit into ecosystem scale processes. The example I give here is work by Morgan Steffen, Tim Davis, George Bullerjohn, Mike McKay, and others, looking at viral infection of large microcystis blooms in Lake Erie, where we think that viral infection actually helped the toxins escape the cells and get into the water supply that caused the shutdown of water in Toledo, Ohio for 400,000 people on an August weekend in 2014. More so, we know that viruses don't just kill things. They help things live, they help things evolve. Curtis supervised an experiment while I was there with Marcus Weinbauer where we took the viruses away and everything stopped growing. And we've seen this type of response time and time again. In Lake Taihu in China, we've been able to show that cells can live at very high densities because they become lysogenized and appear to be resistant to superinfection. Other things we've seen in a paper led by Eric Zinser and David Talmy at the University of Tennessee is that viruses can create memory in cells. And when we look at all the toxic freshwater bloom formers, they have incredible numbers of restriction modification systems, yet viruses still find their way around these. And then the last thing I think that I want to mention is that there's more out there to discover. This table is from a paper in 2016. The table was put together by Eric Gann. Eric went through the literature, pulling up old TEM images, finding picture after picture after picture of beautiful algae with virus cells in them. Most of the ones on this list we have great pictures of, but we've never come close to isolating a virus. So there's a lot of work out there for young scientists and the next generation of researchers. And as we find new viruses, we come up with new answers. And then finally, and this is probably the most important thing to me, what the Viral Shunt did for me is it gave me an opportunity to work with amazing collaborators, make a lot of good friends. What you're looking at here is word clouds of co-authors from the Suttle lab and the Wilhelm lab. I blatantly stole this idea from Philip Boyd's presentation 2 years ago. But just a thank you to all of them for believing in viruses over the last few years. And finally, thanks to the current people we have, I'm really happy that my lab is fully vaccinated and we're all back to working together. And I would be remiss if I didn't mention the funding agencies that have supported virus research in aquatic systems over the past 20 or so years. Thank you very much, and I'd be happy to try and answer questions.
Thank you, and congratulations to you both. We'll give people a moment to add questions to the chat.
And Curtis is here, so I can defer all questions to him, of course.
So far, it's all praise. There's often the notion of using, oh, they're moving too fast. There's often the notion of using viruses in restoration efforts. Any thoughts on where that is?
It's by the president. I better get this one right. If you mean restoration efforts in terms of constraining harmful algal blooms, was that specifically the question, Roxanne?
Yeah, well, you used to talk about this, and now we're seeing that they may be responsible for releasing more toxins. And, you know, there's still a whole unexplored area of what they could potentially be used for in these types of efforts. And in, I would say, the beginnings, this was kind of one of the ones that we were considering. So I just wanted to, if you had anything to say about that.
Yeah, a lot of the work in the 70s and 80s in freshwater, looking at cyanophage, people like Martin and Fox and others, they thought they could use viruses to constrain freshwater blooms. But what you end up doing generally is just selecting for resistant genotypes that will bloom anyways. And in fact, when we look at places like Lake Taihu in China, it seems the majority of the cells are already carrying a viral infection, which is baffling to me, but it makes sense if that infection is at least getting them through the season.
I wanted to just make one other very brief comment, and I think Steve would agree with me. I just wanted to also acknowledge someone else who's on here, which is Amy Chan, who was also there at the very, very beginning and is still a part of my lab. She's also my spouse as well. But from the very beginning, when we started the lab down in Texas, and through the whole process, and still, Amy's still there, kind of often behind the scenes, but also very much a person that's from the very, very beginning sort of started the whole viral concept when we set up the lab together in Texas. And one of the stories that I frequently tell is that when we submitted our first paper to the National Science Foundation, maybe Phil Taylor was there then, I have no idea, but it kind of came back and it said, we have nobody to review this paper because we've never seen a paper on, or sorry, a proposal on viruses in the ocean before, and we can't actually find anybody that works on viruses in the ocean. So it ended up going half to virologists and half to oceanographers, both groups, which hated it terribly because virologists said we can't work on viruses, we don't know what they are, and oceanographers said we don't really want to know what viruses are. We just want to know what they're doing, right? So it's an interesting, so it's nice to see how that evolved from just a few people when, as Steve said, we went to meetings, there was only like 12 of us in a room, to now having meetings where we're sort of acknowledged, and finally it's getting into the textbooks and things like that. And all of the trainees that we've had, and Steve mentioned all the co-authors, but I think what's really impressive is the number of people that have gone through now that are setting up labs all around the world, and have set up labs and are also generating their own graduate students and postdocs and professors as well. So it's really, really a fun time. Yeah, so Phil, that was an interesting story. Going back to NSF, they eventually did fund me, and I'm going to dig out these reviews somewhere if I still have them, because we only had 2-year proposals at that time. In that 2 years, we ended up with a Nature paper out of the first one and a bunch of ones. Then we went in and submitted for renewal, and I could never get renewed by NSF. And I still remember one of the reviewers saying that that they've certainly done a good job ensuring that there's lots of viruses in the ocean, that they're important, and I think the book is kind of closed and we really don't need more. And then so for 3 years, I was unable to get any funding through NSF, and eventually we did again, but it was actually the organization that actually funded the program for a decade was actually the Office of Naval Research, and they came in and essentially for 10 years were our major funding agency. But no, I had a lot of trouble with NSF. It's that usual thing, they want so much preliminary data, and of course we were just kind of, I was an assistant professor and just kind of driving along. So anyway, so I want to thank it's people like Steve and carrying, you know, the flag, and the people that have gone through the lab over the years, and Amy, who's been there behind the scenes, and everyone has certainly been, it's been an incredible community. And I think that's what I really love about it. It's very supportive. Everybody tries to help each other. And I think that's why the field has done so well. Thanks, Stephen. Great talk.
Thanks, Curtis. Thanks for the opportunity.
Just a couple of additional questions. Is there any relationship between chemical pollution and marine viruses? I
guess I'll try that one. I think that marine viruses are a natural part of the system. And that's something that, depending on the audience I'm talking to, they don't always know that. Will viruses change because of chemical pollution? Sure, because you're going to change the host populations, you're going to change the interactions between the viruses and the host. Again, viruses are a natural thing. We're living in a world right now where you say the word virus and you scare people away just because of what's been happening. And I've always used to joke that the easiest way to get an empty row on an airplane seat was to sit down, cough, and tell people I was a virologist, and people would just clear out. But viruses, they do more good things than bad things. Maybe Curtis has a better answer than I do for that.
Yeah, I don't really, I mean, I don't know with the chemical pollution, if I got it right, but I mean, I guess, Steve, your example of viruses releasing the toxins from microcystis is certainly probably one of the best examples of direct connections between viral lysis and chemical pollution, in quotes. But I think there may be a lot of other stories like that as well, because a lot of microorganisms contain toxins. And it turns out, again, depending on the organism, and some of the work we're doing right now, hopefully will be out soon, but looking at lysis that's going on from individual species, right? And so it's very, very different across different species. And so you can imagine for harmful algal blooms of eukaryotes as well, where we think viruses are actually really regulating some of those populations. And there's been examples, for instance, it's not pollution, but DMS and things like that being released from algal cells in huge quantities when blooms lyse as a result of virus infection. And so anything that's inside a cell, which is released, which is a toxin or biogeochemically active, certainly could be very, very important. I don't know if that's what you mean. There's certainly lots of examples of things like human pathogens being released in pollution, and all those kinds of things have been known forever. And that's not really chemical pollution. That's microbial pollution from sewage treatment plant and wastewater and things like that. So not sure if that answers the question at all. So, but.
Seems like a great start. Thank you. And then in working with viruses both in the ocean and in lakes, are you seeing interesting similarities or differences?
Steve, you go with that one for sure, because you're doing lots of lake and ocean work.
There's a lot of viruses in both places. That's the big similarity. I think that thinking about differences is hard. I would say that there's more similarities than differences. The other comparison I could make would be to think about working in coastal versus offshore ocean systems, where we see tremendous differences. And a lot of that is in some ways dictated again by the biology that's there that can serve as a host for the viruses.
The one thing that's a little bit different, but is really, really cool, is that the viruses in fresh water and the ones in the oceans are actually, they have the same relatives in different phylogenetic groups, but they're also really different. And so for some of these viruses, like the algal virus and things like that, you can see where the freshwater versions have originated from the marine counterparts many times. And so you see when you do phylogenetic analysis of different kinds of viruses, for example, you'll see the tips of the branches are all the freshwater ones have come from a marine origin, but often multiple times. So you'll see, for instance, with the phycodnaviruses, which infect eukaryotic algae, for example, you'll see where they've colonized freshwater systems maybe a half a dozen times based on where they've come out of the marine counterparts. And then once they're in the fresh waters, they're globally distributed because viruses are not dispersal limited largely. So it's really cool. So you see these phylogenetic branches emerging out of the marine counterparts. And so one of the things I think that's, again, there's been lots of connections with other parts of science, again, which has made this so cool. But when we isolated the first RNA virus infecting a proteus from seawater, at that time, everybody said RNA viruses were recently evolved because they only infect higher organisms. And now it turns out that we can see roots for all viruses, essentially from marine microbes, eukaryotic and prokaryotes. And it's pretty clear that as these organisms transitioned from the oceans, they just followed their counterparts onto land and into freshwater systems. And the viruses did the same thing, like they do evolutionarily. They're absolutely dependent on their hosts for infection. And so you see these long branches where the viruses have essentially emerged, tracking their hosts as their hosts move from marine into freshwater systems. So it's kind of cool that way too.
And, you know, I would add to that, Curtis, the story of the Synechococcus phage, where we can use a marine host to isolate viruses from fresh water still. And they have many of the same auxiliary metabolic genes, like the photosynthesis gene, but those genes are phylogenetically absolutely unique, which is just baffling to me. So it means that they separated a long time ago, but they've carried the same genes with them.
Yeah.
Very interesting. Clearly, there's a lot of interest in viruses in the aquatic ecosystem. And I thank you both, or your teams, for exploring this area and really bringing it forward to us all and expanding on these.
Yeah, thank you, everyone. We're just starting. So all those other folks that want to jump on the virus bandwagon, there's lots to do. I wouldn't call it a bandwagon. I would say it's fundamental science, which is really kind of changing our ideas into how many ecosystems work at everything from charismatic megafauna, where we're finding all kinds of new viruses, which are involved in population declines and things like that, to what's going on from like the Shunt paper, which is affecting microbial communities. So thank you. Thanks, everyone. Thanks, Stephen. And over the years, it's been fantastic. Thank you.